Hello, everyone. My name's Jacob. My partner, Katie Davis, and I are going to be presenting on the P1Pk and globoside blood group system. Now, there's a lot of info to cover in this presentation, so I'm just going to briefly highlight the objectives that are unique to this system. You are, however, responsible for all the objectives listed on the slide. Feel free to pause the video and look over the other objectives. All right, here we go. In this video, we're going to be talking about how P1Pk is involved in numerous disease states, such as shigellosis and chronic UTIs. We will also talk about how P2 phenotype individuals are able to form non-RBC immune antibodies to the P1Pk phenotype blood. There are also specific transfusion reactions we will discuss, such as the severity of the P1Pk antibodies, as well as the antibodies associated with paroxysmal cold hemoglobinuria. The history of the P1Pk blood group system starts in 1927, when Karl Landsteiner and Philip Levine characterized the P blood group system. They were performing experiments that involved injecting human red blood cells into rabbits to look for new antigens via the detection of antibodies that formed against them. Over time, researchers discovered more antigens and antibodies, adding to the complexity of the so-called P system. As the biosynthetic pathways and genetics of the antigens were further described, it was shown that some of the antigens in the system were quite different from each other. Thus, the P system was split into two blood group systems, the P1Pk and the globoside system. Okay, here are the antigens that we're going to talk about in the lecture today. So we have our P1, PK, NOR, and P antigens. Now, in reality, there are actually three separate NOR antigens, NOR1, NOR intermediate, and NOR2. So I'm not sure why multiple references only generically label NOR as one antigen instead of differentiating between the three. I'm just going to have to assume that someone smarter than me knows what they're doing. There are other antigens that are related to this blood group system, such as the Luc antigen, PX2 antigen, and the FORS antigen. But for the sake of time, I've chosen to omit these. Okay, so I know this chart looks intimidating, and unfortunately this is actually a simplified chart of the biosynthetic pathways involved in the P1Pk globoside system. The good news is that there's actually no need to memorize it. There are a couple things I do want to point out to you though. Both P1Pk and globoside blood group systems do have a common precursor. That's the lactosylceramide. On the left is the pathway involved in forming the P1 antigen, which is built off a type 2 precursor molecule, and that's in the P1Pk blood group system. On the right is the pathway that involves the production of Pk and P antigens, and those are in their respective blood group systems. Notice how the P1 antigen and Pk antigens are part of the same blood group system, despite being part of two separate pathways. This is because the enzyme responsible for synthesizing both antigens is produced by the same gene. Here's a drawn-out picture of all the antigens we'll be talking about in lecture today. There's no real need to know this. I just thought it'd be cool to see the molecular structure of the antigens. One thing that is characteristic of the P1 antigen is that its expression varies from person to person. The dosage effect is partially to blame for this scenario, as homozygous P1 will react stronger than heterozygous P1. However, since life is never that simple, there's much more to it. For example, as we'll mention later, P1 antigens are poorly expressed in children under the age of 7. So age plays a factor here. Also, random mutations in the promoter sequence of the gene is also responsible and can also play a role in the expression of P1 antigens, but that has yet to be fully discovered or elucidated. Erythroid cruppel-like factor 1, also known as EKF1, is a gene that codes for a zinc-binding protein that acts as a transcription factor, and when it binds to DNA, it will enhance the transcription of the associated DNA. EKLF1 is important for the biosynthesis of many blood group antigens, including P1. Loss of function mutations leads to reduced or non-existent expression of these antigens. Phenotypically, this is known as the inhibitor of Lutheran gene, as it was first noticed and described in the context of the Lutheran system. The gene responsible for producing the P1 and PK antigens is called A4-GALT, which codes for the enzyme alpha-1,4-galactosyltransferase, and this is located on chromosome 22. As we will see in the next slide, PK is known as a high-incidence antigen, being present in essentially all individuals. Yet, P1 is present in approximately 80 to 90% of individuals, despite being coded for by the same gene. This is one of the many enigmas of the P1Pk globocyte blood group system. No one really knows the answer as to why this is the case. A study in 2010 did theorize that the discrepancy could be caused by a modified reading frame and the promoter sequence of the A4-GALT gene, therefore leading to a phenotypic P1 versus P2. But the study failed to show any preferential bias to suppress the P1 antigen while still allowing the PK antigen to be produced. On another note, a GC substitution mutation on the 631st position of the A4-GALT gene switches a glutamine for glutamic acid, making a mutated alpha-1,4-galactosyltransferase that has a lower specificity. This lower specificity gives the enzyme the ability to continue to add galactose onto pre-existing antigens, such as the P antigen. The modified P antigens are known as NOR antigens. These antigens are actually pretty rare, and they're really only seen in certain regions of Sweden and in the Amish population. It does seem overkill to be talking about antigens that have such a low frequency, but they are important because all individuals who do not have NOR antigens will have a non-RBC immune NOR antibody that will act as a polyagglutinin in testing. Okay, there's not really much to say about this slide. It is important for you guys to know this information, or at least be able to recognize a pattern that you can come up with the information yourself. But I don't really have much to say about it because we've either already talked about it or we will talk about it. If you feel so inclined, you can go ahead and pause the video so that you can write down this information in your notes. It will help with a couple of the quiz questions. If for some reason you thought that the P1Pk globocyte system couldn't get any more complicated, well, you'd be wrong. During fetal development, red blood cells will weakly express P1Pk antigens until 12 weeks of gestation. Then, the antigens will become undetectable for the rest of pregnancy. After birth, antigens in the P1Pk system are then unreliably expressed until after the age of 7. In which after age 7, their adult phenotype is usually expressed. While no one really knows why these antigens happen to disappear, one theory suggests that it's caused by the downregulation of the EKLF1 gene. And as it turns out, EKLF1 gene and fetal hemoglobin are inversely related to each other, providing a physiologic need for the gene to be suppressed. Because P1Pk antigens are poorly expressed on fetal red blood cells, one might assume that antibodies to this system would not cause hemolytic disease of the fetus and newborn. That is correct. However, the placenta is dense in P1Pk antigens, and it can still cause spontaneous abortions. So while it doesn't cause HDFN, it is still clinically significant because it does cause a pretty serious condition. Here we will talk about the P1Pk antigens and their physiological function. While I wasn't able to find a normative function for the P1Pk antigens, it is implicated in quite a few disease states. As it turns out, many various pathogenic bacteria use proteins that bind to the antigens found in the P1Pk system. These proteins are creatively named P-adhesion proteins, or PAP for short. The most notable of these bacteria is uropathogenic E. coli strains, which can adhere to epithelial cells of the bladder and cause recurrent UTIs. The PK antigen, as it turns out, is the receptor that is targeted by Shiga toxin, and when it binds, it causes the Shiga toxin to be taken up by the cell. From there, the Shiga toxin will be allowed to wreak havoc on ribosomes of the cell. Anecdotally, we can assume that individuals with the P-null phenotype are resistant to shigellosis and Shiga toxin producing E. coli. However, when I was looking for this, I wasn't able to find anything that stated one way or another. What I did find instead is that people with Fabry's disease are resistant to Shigella. Fabry's disease is a deficiency of lysosomal alpha-galactosidase. Amongst other carbohydrates, PK antigens can't be broken down in the cell, and they'll instead accumulate in the cells. The high PK expression in these cells that usually have a low expression of PK antigen now become the target of Shigella. But in most instances, the normally low PK expressing cells are resistant to Shiga toxin.
Hey everyone, it's Katie, and we're going to talk about some antibodies that you might encounter in the P1Pk and globocyte systems. First up is anti-P1. Anti-P1 is a common antibody in P1 negative patients. About 67% of P2 patients have an anti-P1 antibody. This antibody is naturally occurring and non-red blood cell immune. Usually anti-P1 is not clinically significant, but it can cause both acute and delayed hemolytic transfusion reactions. These reactions can be quite severe. Unfortunately, this antibody can show variable reactivity with different cells and can be easily missed in screening and panel testing. Anti-P1 antibodies are usually IgM, but they can also be IgG. Usually only the warm reactive IgG antibodies are clinically significant, but there have been weird cases that prove as exceptions to this rule. This antibody has also been shown to display variable thermal amplitude as well. In one case study I read, they were having a hard time figuring out what antibody was causing the reaction because they didn't see any reactivity when they did the screen, and the cross-matched blood looked compatible in the lab, but then the patient started to have a transfusion reaction after they began the transfusion. What they ended up finding out was that the antibody that was causing the reaction was an IgM antibody that only reacted in the body temperature and didn't react at all in the cold, which is why they missed it in the lab. And it also didn't react with the AHG phase of the IAT as well, which is pretty interesting. Anti-P1 is not implicated in HDFN for a couple of reasons. Usually the antibodies are IgM, which cannot cross the placenta, but in the cases where it is IgG, it cannot really react as the fetal cells have poor antigen expression and there is nothing to bind to. Some sources of naturally occurring P1 substance include hydatid cysts from the Echinococcus granulosus tapeworm, pigeon blood and pigeon droppings, various liver flukes, which are the trematodes or flatworms, and also egg whites. Higher titers of anti-P1 antibodies are commonly found in people who are frequently around birds or who work with birds. Pigeons are the birds usually associated with P1, but it has been found in several other types of birds as well. These substances can actually neutralize anti-P1 in vitro testing if the panel testing shows complex or confusing reactivity due to multiple antibodies. Anti-P1Pk, which is formerly known as anti-TJA, is an antibody found in people with the extremely rare little p or p-null phenotype. These patients lack all P, P1, and PK antigens. If you remember from earlier in the presentation, P and PK antigens are found in nearly 100% of the population, and P1 is found in the majority as well. So this antibody is extremely rare, but is very clinically significant. The antibody is non-red blood cell immune and will react with every red cell except those of the little p phenotype. Anti-P1PK can be both IgM and IgG and shows high thermal amplitude, meaning that there is a very large temperature range in which it efficiently causes increased red cell destruction. It is very clinically significant, and transfusion with incompatible blood will cause an immediate febrile hemolytic transaction and will likely be fatal for the patient. Because anti-P1Pk often has IgG components, it can cross the placenta and cause severe HDFN. It can also cause spontaneous abortion, usually within the first trimester. It is very efficient at binding complement, and complement-mediated hemolysis is to blame for the rapid destruction of incompatible red cells. This antibody can be difficult to identify as it will show reactivity with all panel and screening cells, but not with the autocontrol. It will likely cause ABO discrepancy as well, with extra reactivity in the reverse type. Allo-anti-P is a naturally occurring non-RBC immune antibody produced by the individuals of the PK phenotype. This antibody will react with all cells except the autocontrol and cells that are either little p or PK phenotype. Both of these are very rare as almost everybody has P antigens. The person who makes an allo-anti-P antibody may type as P1 positive. This antibody can be IgG or IgM and is very clinically significant. It can cause hemolytic transfusion reactions, HDFN, and early-term abortions. P is an antibody directed against a person's own red cells, which contain the P antigen. Remember that approximately 100% of people have P antigens. This antibody is found more often in children than adults and usually follows a viral infection. However, when it does occur in adults, it is usually the result of chronic relapsing episodes of tertiary syphilis. The main thing to note about this antibody is its association with paroxysmal cold hemoglobinuria, or PCH. PCH is an autoimmune hemolytic anemia. It is usually self-limiting, but sometimes can be severe enough that the patient will need a transfusion. Auto-anti-P is a cold-reactive IgG biphasic hemolysin. This means that it binds in the colds, and then it lyses cells when it reaches body temperature. The Doneth-Landsteiner test, named for the guys who discovered it, is used to confirm PCH. This test involves cooling the patient's sample to 4 degrees to allow the autoantibodies to bind to the P antigens on the red cells. The sample is then heated to 37 degrees, where complement will be activated and hemolysis of the red cells will occur. Anti-Nor is an extremely common antibody found in most people's sera. The Nor antigens to which they bind are very rare and have only been identified in a few families worldwide. These Nor antigens are part of the P1Pk system. Nor cells are considered polyagglutinable as they react with most human sera. Anti-Nor can cause problems with transplants and transfusions in these Nor-positive families. This can include anything from an organ transplant to a skin graft to a regular blood transfusion. Thanks for watching the presentation, guys. So these next couple slides have our references. We also have some quiz questions coming up as well to help review some of the material and to give you guys a little bit of practice because this system is one of the more complex and difficult ones to learn about. Lactosyl-ceramide. And then if you remember back from when Jake was explaining the biosynthetic pathways that these antigens follow. What phenotype is expected to have the most amount of PK antigen on the red blood cell? A. P2, B. P2K, C. P0, or D. P1K? This is B. P2K. So P2K is going to only have PK antigens present, and so they will have a lot higher number than they would in cells that share other antigens as well. 3. Which of the following units would be compatible in a patient with anti-P1Pk? A. Group O P2 phenotype, B. Group O P1 phenotype, C. Group OH or Bombay P2K phenotype, or D. Group O little p phenotype. Answer is Group O little p phenotype. Anti-P, P1Pk will react with everything except the little p phenotype due to antigen presence. And then this table is just a quick review of which phenotypes are associated with which antigens. Or, which of the following antibodies is associated with paroxysmal cold hemoglobinuria, or PCH? A. Alloanti-P, B. Autoanti-P, C. Anti-P1, or D. Anti-P, P1Pk. This is autoanti-P. Question 5. Which of the following is not a known source of natural P1 substance? A. Goat's milk, B. Pigeon droppings, C. Hydatid cyst of the Echinococcus granulosus, or D. Liver flukes. This one is A. Goat's milk. So you guys are safe to drink goat's milk and not develop antibodies. Thank you guys so much for watching our presentation, and I hope you were able to learn a lot from it.